All right, this is Notes on Enzymes. Enzymes are very important for chemical reactions. There are two general parts of every chemical reaction. We have our reactants, and those are the chemicals that participate in the reaction. Think of them as our ingredients. And then we have the products, the substances that are formed as a result of the chemical reaction. In this case, we have our reactants of the sample equation, and this is actually glucose, C6H12O6, and when it is reacted with oxygen, the results are carbon dioxide and water. This is cellular respiration. So where do enzymes fit in? Enzymes are proteins, so one of our biomolecules, that catalyze the rate of a chemical reaction. And to catalyze something is to speed it up. Enzymes speed up reactions by decreasing the amount of activation energy required for a reaction. Activation energy is the energy that chemicals need in order to begin the reaction. You have to put a certain amount of energy into a system in order for the chemical reaction to occur. So here's an example, a visual example using frogs as opposed to chemicals. So in this example, I have frogs leaping over a pile of rocks. The higher the barrier of rocks, the less frogs can get over. And think of this as being less product being created. If I remove some of those rocks from the barrier, that's basically what an enzyme does. And it allows more reactants to make their way into products. So enzymes reduce the amount of energy I need to put into a system in order for a reaction to occur. Without enzymes, typical everyday reactions that occur in our bodies would occur far too slow for us to survive. We would not be able to break down toxic wastes in a timely manner, and we would not be able to metabolize our food fast enough to have energy to power ourselves. Life cannot exist without enzymes. So this is a graph that shows the basic relationship between energy for reactions and how enzymes affect that. So I have my reactants, and I need to increase the amount of energy in order for the reaction to occur. If I add in a catalyst, so that's our enzyme, it reduces how much energy I have to put into the system in order to have the reaction and get my products. Some examples of enzymes. So first of all, I want you to look at the end of each enzyme's name, ACE. That's important. So I have carbonic anhydrase. Carbonic, think carbonation or carbon dioxide. Carbonic anhydrase is found in your blood. It speeds up the removal of carbon dioxide from our bodies. And since carbon dioxide is bad for us, this is a good thing. Catalase, it's found in liver tissue. It speeds up the decomposition or the breaking down of hydrogen peroxide in our bodies. And amylase, it's found in your saliva, and it speeds up the breakdown of sugar for digestion. It gets those starches broken down into simple sugars. So you notice all of these enzymes have that ending of ACE. When you see ASE at the end of a term, that's going to be an enzyme that breaks things down. And we have the substrates or the reactants that those enzymes work with. The enzyme substrate complex. So the substrate for an enzyme is a molecule that attaches to the active site. So it's what the enzyme is working on. The enzyme and substrate are specific to each other, like a lock is specific with a key. So the catalase substrate, which is hydrogen peroxide, that's what it acts on, would not attach to amylase. They won't have the right shape. Once the substrate and enzyme bond, it becomes the enzyme substrate complex. So if we look at our example with a lock and key, so they'll think of the lock as the enzyme and the key is the substrate. So when the lock has the key fitted into it, this is the only key that's going to fit in this lock, it becomes the lock-key complex. When we have enzymes, we have our enzyme here is like the lock. It has an active site, a specific shape that the substrate fits into. And this shape is specific to that substrate. And then once they're together, we have the enzyme substrate complex. After the chemical reaction, the product, which started out as my substrate and has now been acted on, is released from the enzyme. The enzyme is free to carry on the same chemical reaction again and again because enzymes can be reused. You can use the same enzyme over and over and over. That's very important. So here is my enzyme, and this is the active site, and this is my substrate, so what the enzyme is going to bind with. Enzyme substrate complex, so the enzyme has bound to the substrate. It acts on it and breaks it apart. In this case, this must have been an ACE, ASE enzyme, and it has broken my substrate into the products. There are some factors that are important to remember when you're thinking about enzymes, and there are things that can make enzymes far less productive. And those two things are pH and temperature. So think pH is like the acid-base scale, and there's a certain range of pH that enzymes are happiest in. And then temperatures, there are certain temperature ranges that enzymes are happiest in. If the environment, which in this case would be the human body, is altered, the enzyme activity may decrease. So for example, when people have high fevers and they pass away or they die, it's not just because their bodies are at a very high temperature. It's because the enzymes in their body cannot function properly because the temperature is too high.